Um, Wonderful. Well, thank you, Clarissa and Zach, for having me. It's really an honor to be part of this program. And thanks to, I can see you all there. Thank you so much for coming in and being willing to put up with a virtual talk. I'm normally based at Friday Harbor Labs, but right now I'm working at Hopkins Marine Station in Monterey, so it would have been a bit hard to get there. And so I would like um, to point out that we do all of our work, most of our work, we're actually working coastwide from San Diego to Alaska on actually both of the projects with the eelgrass and the sea star wasting. But our ground zero for research effort is Friday Harbor Labs. And I just wanted to introduce some of the key people in my group. Postdocs Becca Mayer and Olivia Graham, and graduates, PhD student Grace Crandall, based at Friday Harbor Labs. Since most of our research is done in the San Juan Islands, where the Coast Salish people have stewarded these lands and waters in the past and to this day, I would also like to acknowledge the tribal support for co-management of these waters. And the projects that I'm going to talk about today, we're ecologists largely, although we work kind of at all levels of biological integration. But both the Sea Star Wasting Project and the Eelgrass Health Project are continental scale projects that have been affected by climate change. And so I want to start briefly with the eelgrass to give you a little bit of the background of work we've been doing on that, and then spend most of my time talking about the sea star wasting. So our team are a group of what we call disease ecologists. We work on host pathogen interactions in the ocean. And as ecologists, we think of these as sentinels of change in ecological interactions. But actually, as more recently, we're finding these are drivers of continental scale community change. And a lot of times it's facilitated by that little red triangle at the interaction of how the host and pathogen fare in their environment with climate warming. We've been finding warming conditions can facilitate at least these two disease outbreaks. And so kind of synergizing some of the regular effects of climate. So I'll try to highlight that today in my talk. So we teach a range of courses at Friday Harbor Labs, and my team has taught a marine disease ecology course since 2012. And we started our eelgrass disease ecology project in 2012 because we had come from working on coral reefs where temperature-related disease agents were causing trouble. And we could tell because what was known about the disease of eelgrass, that there was the same possibility for eelgrass. And so I'm going to give you just a brief snippet of that project, but the slightly sad news is that a decade of our research does confirm that heat waves have triggered major outbreaks of disease and declines in our eelgrasses. And I think everybody in the room, I'm guessing, understands the importance of eelgrass, particularly in our Puget Sound and Salish sea waters. I'm not going to say a lot about the benefits of eelgrass or seagrasses because I think everybody knows that they're vital nursery grounds, vital foraging places. So I just want to tell you about one extra, what we call one health co-benefit of seagrasses, including our eelgrass, and that's reducing pathogenic bacteria to humans and wildlife. And this is work we started in Indonesia. But most recently, this paper led by Dawkins is about Puget Sound. And the work that we did in Indonesia, you can see these pictures here. How's the volume, by the way? Can everybody hear me okay? Maybe give me a thumbs up. Oh, good. We actually have audible. That's good. So what we found is inside our seagrasses in Indonesia, there were very few of these red spots, which are an indicator bacterium. But just outside, across 4 different islands, the levels of bacteria, the same species, were extremely high. And so there's a whole paper that goes with this that I'm not going to talk about, other than just to make the point that we provided evidence that the seagrass meadows are actually diminishing the levels of pathogenic bacteria. And recently in 2024, we were able to find similar results for bacteria in Puget Sound. And so this reminds us that there's really important services of our natural ecosystems. And we're even thinking that the value of eelgrass can be valuable to our sea stars, and I'll come back to that. Okay, but our eelgrass or seagrass needs to be healthy in order to provide these kind of services. And we've been studying a disease agent called Labyrinthula zosteri. I'm going to probably call it Labby for short. And this is the kind of lesions that it creates. And for those of you that are out and about in your eelgrass meadows, you'll be able to find these if you just pick up a few blades of the eelgrass and look for these kinds of characteristic lesions. That's what's caused by this protist. It kills plant tissue, it reduces leaf growth and sugar reserves, and impacts the survival of the plants. And so our team has produced over the last decade probably 20 papers sort of around this theme in our waters. And this is what it looks like. So this is what that protist looks like. It has these spindle-shaped cells, and they lay down these mucus networks, and then inside the plant, so actually these are the spindle-shaped cells inside the plant, and then this is the little bit of the mucus network that it's doing. And our team, let's see if I can get this to change. This is an example. We've done so many ecological surveys, actually from San Diego, whoops, all the way to Alaska, but this is one of the ones in the San Juan Islands that Olivia and Lillian are running. And basically we survey 30 to 50 plants on one of those and then run triplicate transects. This is actually my front yard in the San Juan Islands. This is False Bay, and we have a nice eelgrass meadow there. But what we found, we started this work in 2013 when there were hundreds of plants in our meter squared quadrats. And rapidly over time, these numbers have diminished to quite low levels, fairly low densities of our plants. These are intertidal eelgrass across essentially most of the main islands in the San Juan Islands. At the same time, this shadow box here shows the blob, which was a heat wave that we had for several years, beginning in 2015. And what you can see is this bottom shows the levels of disease in these plants. So we check each plant for the disease, and I'm not going to go through all the diagnostics, but we've spent a lot of time doing the molecular work to identify it. What you can see is that it peaked to the point where almost every plant in our transects had the disease, and the numbers have diminished. And just to give you a picture of what this looks like, this is one of our sites. In 2017, this is from drone footage. So the green is all the eelgrass. This is intertidal eelgrass now. There's also a subtidal bed, a sort of lower, deeper water. These are the 6 transects at that site, and you can see that there's eelgrass on them. By 2021, following another heat wave, we lost all of the intertidal eelgrass, and it pushed way down into the deeper subtidal. This, unfortunately, has happened at virtually all of our intertidal sites in the San Juan Islands. And working with the state eelgrass scientists, we're finding that the San Juans are showing some of the biggest declines in intertidal eelgrass in all of our Washington state waters. So these plants are impacted by a combination of warming and disease. And given the impacts in the San Juans, we've also extended our work from San Diego. These are nodes where different team members have worked all the way to Alaska. And in this work showed that the levels of disease were sensitive to warming across latitudes. And so that's pretty much all I'm going to say. I'm really just giving you a snapshot of that before moving to talk about sea star wasting. Some of the answers we have: does eelgrass wasting disease drive big declines? It certainly is implicated in them. Warming, it's working hand in hand with the warming. We've learned a lot about how this disease is transmitted. It's actually an interesting one. It is waterborne, so it's spread widely in the environment. This is some of those spindle-shaped cells actually colonizing the outside of a plant and getting in through these little boreholes. And the challenges for us are to figure out how we manage for eelgrass resilience through these warming events. One of our findings is that the deeper meadows are apparently refugia, that the plants are much healthier, and so that really puts the focus on some of these deeper meadows for preservation as being very important. So I'm going to move on because I think Clarissa asked me to talk primarily about the work that we've done with sea star wasting disease, but I'll be happy to take questions later about the eelgrass. I'm guessing pretty much people in the audience know that this was an enormous wildlife epidemic. It started in 2013, between 2013 and 2015, depending where on the West Coast you were. It continues. We just had a meeting yesterday of the Sunflower Star Recovery Program, which is about 60 scientists, and there still are reports of wasting. The thing that was quite spectacular about this epidemic is that there were at least 20 species affected. I'm showing here the sunflower star, and I'll get to it in a minute why I'm focusing. Well, that's why. Because we had such a huge decline in the sunflower star. In the process of studying this epidemic, pretty much right away it became clear that the sunflower star was the most susceptible of all the species that were affected. So things like our ochre star were certainly dying in the millions, but there were also a proportion of them were surviving through these events, whereas the sunflower star was just absolutely hammered throughout its range. And it is an endemic species, so it lives nowhere else in the world than our West Coast. We've published a few papers kind of in the past. This is a 2019 paper where we warned that there was the danger of a continental scale collapse of the sunflower star. I think it was a fortuitous paper in that it did bring a lot of attention to this. Before that, though, we were able to rely on citizen science data to show some big declines. And I just want to highlight some of these early findings of, earlier in the epidemic, because it's sort of prescient for what we're seeing now. So there's a bunch of lines on this. There's our sunflower star, and this is reef check data from citizen science divers that goes back to 2006. So we were able to find that the sunflower star was doing really well up until 2013, when bam, they just plummeted and have not come back for the most part. There's other lines here. I want to focus on this one. These are the urchins, and part of the reason why this particular mortality event with the sunflower star has been very important ecologically is because when the sunflower star was reduced, its prey boomed. And we're now having problems with urchins throughout the range. And of course, they're decimating the kelps. And then here's one other line to point out. This is the leather star. And I hope those of you that get out into the intertidal and tide pooling have seen it, because that one actually increased. And I'm going to come back to that at the end and talk a little bit about what it means to have a species that may be resistant in the face of this one that is so susceptible. Is everybody following? You hearing me okay? Everything good there? All I see is kind of dark in the thing, so it's hard not being there with you. So the impacts of the sea star wasting disease, the sunflower star loves to eat urchins, way more than any of us realized before this outbreak. And urchins love to eat kelp. So this is actually showing a macrocystis, which is a more outer coast kelp. But of course, the story holds true for our nereocystis, our bull kelp. And so when these urchins exploded with the removal of their predator, they've decimated these kelp meadows. And we get situations like this that's described as an urchin barrens, where this purple urchin here has taken over. I don't have time to really talk too much about the interaction between the sunflower star and the urchins, but it's ending up being a really interesting story, because if you just bring a few sunflower stars out to a place like this, and some of our colleagues in British Columbia have been doing this, and in Oregon, these urchins will scatter. And so not only does the sunflower star eat them, but it also kind of herds them because they have a fear response to it. And so it's particularly effective in controlling them because of that reason. So because of its ecological importance, the Nature Conservancy has been supporting a sunflower star recovery plan. One of our first steps was to go ahead and get this listed as the Pycnopodia, the sunflower star, listed as an endangered species with the International Union for the Conservation of Nature, which helped sort of create all of the data showing how diminished this species was. Our brilliant colleague at Friday Harbor Labs, Jason Hoden, was able to develop captive breeding. And I like to say, although this was definitely Jason's effort and brilliance, he was standing on the shoulders of giants, because one of the areas of expertise at Friday Harbor Labs is invertebrate embryology. There's not two places in the world where that's the claim, but at Friday Harbor Labs, we teach an invertebrate embryology course most years and know how to breed all kinds of different invertebrates. So Jason was able to tap into that knowledge. And basically, he has outsourced his methods to all of the West Coast aquaria, which are now starting captive breeding programs. And then our project was the epidemiology, figuring out what was the causative agent. And I'm really excited to be able to talk tonight about this result. This is a paper that we published last summer. It came out in August. And this is our team led by Alyssa Gaiman at the Hackeye Institute in British Columbia. So this is a trans-border collaboration between the US and Canadian scientists. Grace Crandall, our PhD student, Melanie Prentice, the postdoc, and me, the disease ecologist who'd been working on this for a few years. And so I'm going to tell you a little bit about how these experiments were done, because they're pretty tricky to do well, right? You may well say, why did it take you guys 10 years to figure this out? And I would like to point out that this work is really needs to be carefully done. It's painstaking, and it often does take 5 to 10 years to figure out an infectious agent in the ocean. So the way this is done is tissues are taken from a diseased star. They're ground up in some way, or maybe water is taken from water close to a diseased star in the preparation of an inoculum, which can then be injected into a healthy star. And then I kind of messed this up a little bit. This is the active inoculum from the disease star, injected into a healthy star, which then shows signs of disease. Up here, the reason this inoculum is sitting on the heater is because for controls, we heat killed all of these different treatments. And so the different treatments included tissues from a diseased star, water run past a diseased star, and eventually coelomic fluid. And so I just want to take you through a little bit of this work. So the first experiments were done just to show that the water over a disease star was infectious, that it was transmissible. And so what's shown here is the time and days to the first signs of disease, which is twisting arms in the sunflower star, and then how many days till they started to drop arms, and then how many days till they died. And so it took up to about 2 weeks for that to all happen with individuals that were initially asymptomatic. The raw homogenate from a disease star produced signs more quickly. So you can see that this time to arm drop and death is being reduced. And then one of the real breakthroughs came when Alyssa said, let's try coelomic fluid, which is essentially the blood of the star. And so that pretty much halved the time for the signs of disease to show up, for the dropping of arms, and for the mortality. At the same time, all of the control stars survived. And that was really important to be able to show definitively that these factors were causing the signs of disease. The next huge breakthrough came when a colleague from UBC was able to culture from a 6-star this bacterium Vibrio pectinacida. Now, a lot of work preceded that in terms of analyzing what was it in this inoculum that stood out as a possible causative agent. And it was this Vibrio pectinacida again and again that showed up in the 6 stars, in all the inoculums that were active. Okay. And so that was pretty cool to be able to use the pure culture of this Vibrio at 3 different concentrations, 10 to the 3rd, 10 to the 5th, and 10 to the 7th. And kind of beautifully, they did a beautiful job on these experiments. The time to arm drop diminished as the concentration. So I just wanted to convince you that these experiments were scientifically really carefully done over a long period of time and repeated quite a few times to produce that paper. At the same time, I'm not going to have time to talk about it, but animals in the wild that were showing wasting also had the Vibrio pectinacida, and animals without the wasting didn't have it. And Alyssa and team have now looked at several other species, and it's holding up as the causative agent in some of the other stars as well. There's a lot more work to be done. This is really kind of early stages, but it's incredibly exciting and unlocking a lot of opportunity for next steps. So what would some of those next steps be? The Nature Conservancy is very serious about this recovery plan and moving forward with conservation breeding. And they've done a phenomenal job of teaming with all of the major West Coast aquaria to establish breeding programs. This is a wonderful picture of to remind me, I'm getting carried away, going too fast here. To remind me, one of the other big new discoveries is the ability to cryopreserve the larvae of the sunflower star. And that means that it's possible to breed millions and millions of larvae, cryopreserve them, and bank them for a future time. And so this is one of those cryopreserved larvae. It's pretty cool, being rehydrated and essentially brought back to life. So this will allow success in maximizing genetic diversity of the sunflower star, which, in talking with a lot of other conservation breeding programs like white rhino and salmon, it's really important that the first step is taken to be sure you have enough genetic diversity. And of course, when you've lost billions and billions of your host animals in the wild, it's important to keep an eye on that. And then the next step is to investigate selective breeding for resistance. At the same time, Jason and his team have moved forward. They've been releasing captives in Friday Harbor. Here they've brought these guys out in bins. You can see these are urchin barrens. So these are areas that are dominated by urchins. And already the sunflower star is starting to clear space from these urchins. At the same time, there are experimental releases in California from the Sunflower Star Lab and other groups. And some really exciting work is being done. I hope I'm not going too fast about this, but I wanted to get it all in. Really exciting work to ask, when you release a sunflower star, can you detect its DNA in the water so that you know that it's there? And these samples are pending, and I don't know the answer to that yet. It's very likely the answer will be yes. And then at the same time, we can begin monitoring the Vibrio pectinacida in the water and ask what the seasonal environmental reservoirs would be. For example, is it in the clams or mussels that these stars are eating? Okay, so as part of the conservation breeding program, Grace Crandall has been working on this project for the last 4 years now. She's a PhD student that I co-advise at the University of Washington. She's been working on this project all along. And she got to do a really cool experiment, again with Alyssa and Melanie, the whole same team, simultaneously infecting or exposing the leather star. Remember, that's the one that was resistant, and that's actually been doing really well. The ochre star and the sunflower star. So this is, we consider our most resistant star. The ochre star in some places has been doing pretty well and seeming to tolerate what we know is still Vibrio in the water. And then, of course, our very susceptible sunflower star. And then this is just a snippet early, some of her results. What she'll do with the coelomic fluid from these stars, remember the coelomic fluid is essentially the blood of the star, is catalog which genes are activated by the infection. And so what happened in this experiment, this is the focal star. It was a sunflower star, well, a bunch of them. They all died really quickly. So the green is normal. The yellow is when they started to twist. The red is when the arm dropped, and then the black is when they died. So these, except for one of them, all diminished pretty quickly. They then spread to this sunflower star, which also all died pretty quickly. The ochre stars lasted a little bit longer, and one of them made it all the way through and recovered. It turned green again. So that was pretty exciting, evidence for resistance in this experiment. And here's our friend, the leather star, all the way through. And I would like to point out, these animals were all co-housed together. So during the entire 60 days, this thing was monitored. It was crawling over dead and dying other stars. And so we're pretty confident that it really is resistant. And so Grace, in the spring, will have data on which genes are important, perhaps, in the leather star compared to the sunflower star. And then I want to leave you with a bit of good news before I turn and say a few things about my book that's out. This is some data hot off the press that was just shared with me today from this sunflower star recovery meeting that we had. This is the iNaturalist data on the sunflower star sightings by year in Oregon and California. So this is 2021. Not surprisingly, there were only 3. In 2022, there were a couple more. There were 6. In 2023, there were 75. And this year, the numbers are up. Now, of course, people may be looking more carefully. These are not carefully controlled data. But the good news is that there are a lot more sightings. And so that doesn't mean that we're out of the woods, but it does mean that there's more hope that there are resistant stars out there. And there's really hope for this recovery program. And so everybody's pretty happy about that. There are still wasting star observations. So again, this is our sunflower epidemiology team, and just incredibly beautiful work by these 3 and our colleague Stephen Roberts at UW and Jason Hodin. So just very quickly, a couple of things about warming disease impacts. to keystone species like the stars and foundation species like the eelgrass. We're finding that infectious disease really can change oceanic coastal communities by decimating on a local scale and in some cases continental scale mortality events. The seagrass protist, remember that's what's killing the eelgrass, does cause higher risk and impact with warming. We're also doing some work in Italy and Palau, and so it's turning out to be a problem with other species of seagrass as well. I think everybody's convinced we have evidence for this keystone sea star being driven to endangerment by a temperature-sensitive Vibrio. And then knowing the agents of disease is absolutely vital to helping us develop solutions. We can now, for both of these, monitor in the water and reservoirs the eDNA and understand how they're transmitted, and the hunt is on for resistant strains. So that was a lot. And it's sort of, well, it's a mix of bad news and good news, right? It's exciting showing the scientific progress. It's also worrisome that we're having to deal with these kinds of issues in our vital marine ecosystems. And so I wanted to turn to a more positive view, because actually these are happy days for me, is when I get to go tide pooling or snorkeling in our waters and just marvel at the incredible biodiversity we do have. Even though we're dealing with some losses, we have incredible biodiversity. And a good day in one of our tide pools, we might see any one of these stars. And if you look at Kozlov's Washington-based key, there are over 80 species of sea stars in our waters. So the reason I wrote this book, The Ocean's Menagerie, is really a celebration of our marine biodiversity, and in particular, our spineless biodiversity. So I'd like to take a couple of minutes to just tell you a little bit about that. Oops, where did I go? So I guess a short way to think about this book is I think of it as the superpowers of the spineless, because to me, these animals do have superpowers. They have biology that is so fantastic. It's up there with anything that Superman or other Marvel heroes have. And I don't have time to talk about all this tonight, but these are the chapters, and I'm going to just give you a little bit of a hint about the work in the sponge's pharmacopoeia and the sea slug's sting, and maybe a little bit about the sea star's sticky skin. But each of these are actually adaptations that have important spin-offs for humans, and in a lot of cases biomedical. So I'll tell you a little bit about that. Okay, we've been going for a while. Let's just stop for a minute. Does anybody like trivia? Anybody? Still? Well, here's one. How old are the invertebrates? And I think about this and I talk about it a fair bit in the book, because it kind of blows my mind that we're dealing with such ancient animals on our planet. And I'm not going to ask you for a show of hands, but I bet everybody was thinking, oh, of course they're 600 million years old. They're incredibly ancient. Dinosaurs are only 250 million years old. We think of them as being old. They've like come and gone. We still have these invertebrates 600 million years later. I'm not as old as the invertebrates, but I have been at it a long time. And yes, I do still dive. And I think my obsession with diving research and being underwater definitely had its beginning as a grad student when I got to be a saturation diver. And I lived underwater for a week in this Aquarius habitat right on the edge of the deep reefs in St. Croix. And so after that first mission, I went back and did 3 more. And this is me actually leading my team once I was a professor. And just to give you a sense, well, why would you saturation dive? Why would you live underwater? On one mission, just one of the 4, we accomplished 85 excursion dives from a saturation depth at 60 feet underwater. and a total bottom time of 194 hours over the 10 days of the project. Now, that's not me doing all of that. That's sort of the whole team. But the power of being able to really do work underwater is unbelievable. And so it was pretty exciting. Okay, bio-inspired design. That is one of the themes of this book, and I'm going to just briefly talk about some of the cancer drugs from sponges. Because I was fortunate early in my career to be able to work with natural products chemists, including John Faulkner's group, and they discovered this swinoolide A, which is this really complicated compound. And they were finding it inside the sponge, but it turned out that it's made by the bacteria. And that was sort of the key discovery that's sort of gone on to be found again and again for these really important cancer drugs that are found inside sponges. And some of the activities these compounds have is they're antifungal. Cytotoxic is the big one because it means that disrupts cell divisions. And so that's why sponges have been a very rich source of cancer drugs. So Helicondria panacea, probably everybody knows that. It's actually in our waters. Houses a Streptomyces bacterium. And its synthetic derivative is currently used in the treatment of 5 different carcinomas. Helicondria, a related species of helicondria, houses bacteria that makes helicondrin B, which has been the synthetic version approved to treat breast cancer. A new thing that a friend just sent me is this very sad story. Unfortunately, Tatiana Kennedy Schlossberg just died from leukemia, but she had I have this quote that was sent to me. My plan, had I not gotten sick, was to write a book about the oceans, their destruction, but also the possibilities. During treatment, I learned that one of my chemotherapy drugs, cytobarin, owes its existence to this Caribbean Tectathea crypta, and that's what this sponge looks like. So I could go on, and I do kind of in the book talk about quite a few examples like that. Okay, what about our local nudibranchs? What kind of superpowers might these have? And I'm guessing that probably nobody in the audience really is interested or likes nudibranchs, right? They're not very interesting. I'm hearing a laugh. Good. They're just a sea slug, right? No, they are just amazing, and they are one of my favorite groups. And I did work on them as a PhD student, which of course was a really long time ago. And so I still love them. And so if I was there, I could ask you which of the species shown here is the one that actually does this thing, which is uptake the stinging cells from its prey. To kind of move through this quickly, I'll show you that, of course, it's the Hermesenda shown here. These other ones, while they may eat sponges or some of them may eat cnidarians, do not uptake the nematocysts. So it's only the aeolid groups of the nudibranchs. And of course, the beautiful Aeolidia is an aeolid. And here it is stalking a sea anemone, which is where it uptakes the stinging cells from the tentacles of the sea anemone. It ingests them, passes them through its digestive system and into the projections on its back. Oh, we need you to leave now. Oops. Another species of aeolid is the blue dragon, which I have to say I've never actually seen it, but it's a pelagic nudibranch that feeds on the Portuguese man-of-war. And I've just included this picture of it in glass because my first book was about the Blashka glass invertebrates, of which this is one. But I don't have time to talk about that tonight. But if you're interested, you can find that on my website. But the reason I'm showing the blue dragon is not only do Portuguese man-of-war take down divers, snorkelers, and people in the water, but the blue dragon does too. It not only uptakes the nematocysts from the Portuguese man-of-war, but it concentrates them. And so they had to close beaches for the blue dragon. So how does this work? It's called kleptonidy, and it's what in this chapter I call the superpower. And the nudibranchs selectively ingest the immature nematocysts, pass them through their entire digestive system, and then into these pouches on their backs, which are continuous with their digestive system. And then these are the nitophages, or the developing nematocysts, developing here, run by the nudibranch. It's developing the biology of an organelle from a species across the tree of life. And that is the really remarkable thing about this. And then when these mature, they can fire and close beaches. And then the superpower for the sea stars, and I'm kind of hurrying a little bit now, but I do need to just acknowledge the beauty of this diagram. So each of the chapter openings in my book, these diagrams were done by Andrea Dingledine, and some of you may know her work. She's based in Santa Cruz now, but she's been in Friday Harbor. And so I really love this because it shows the ochre star in the intertidal and the sunflower star subtidally, as well as other ones you may know. And so the superpower that I talk about in the sea stars is their sticky skin. And this is a pretty fantastic one too, because sea stars are able to open clams. None of you or me can open a clam with our bare hands. Sea stars can do it because of the suction in their tube feet, but also their ability to create a stiff turgor in their skin and hold it for hours and hours, and then they basically can outlast the clam. And this is done by cross-linking these collagen fibers and creating stiff skin. And of course, the other magic of this sticky skin in the sea star is the ability to, at the touch of a neuron, disarm the sticky skin and drop an arm. And so that's also how they autotomize an arm. And there's a lot of biomedical applications to this. There's journals written about soft matter for biomedical applications in transplantation surgery. So these are the 8 chapter openings for that book, of the 8 chapters about all of our wonderful spineless superheroes. And then here's my tiny plug for the book. I'm terrible at self-promotion, but I am really proud of the fact that the Wall Street Journal picked my book as a holiday gift book that they recommended, and Nature had a really beautiful summary in their best books in brief. And then just 2 days ago, Earth.org wrote an incredibly beautiful review about the book. So I hope some of you will be interested in having it. And that's, I'm going to end now because I would really love to have time to answer questions. So I'm done. I'll leave it at that. Are there any questions? Happy to answer questions about anything.
Thank you so much. This was really fascinating. I've been getting curious about marine pathogenic vibrios recently at work, and I was wondering, my understanding is that there's an incredibly high diversity, like a bafflingly high diversity of pathogenic vibrios in marine environments kind of all the time. Do we have, and that it can be really difficult for that reason to sort of trace back causative agents. What do we have an idea of what sort of the ambient concentration of the wasting disease vibrio?
Yeah. That's a great question. There are thousands of different species of vibrios, not always all in our waters at the same time, but potentially it's an incredibly diverse strain, series of species and strains of bacteria. Vibrio pectinacida is just one of them. Vibrio pectinacida is not a human pathogen, so you cannot catch it from a sunflower star. But but Vibrio cholerae is the bacterium that causes cholera. Vibrio parahemolyticus and Vibrio vulnificus are human pathogens that are present in seasonally and need to be tested for in our shellfish, in clams and mussels. And so there definitely are many species. Vibrio pectinacida is quite rare in our waters most of the time, although we don't have very good data on it. And that effort to try to understand where and when it's present is really kind of just beginning. It has previously been known, I kind of forgot to say this, has previously been known as a pathogen of marine invertebrates. For example, Pecten acida means scallop killer. And so it was described in Europe as a pathogen of scallop larvae. There have been some episodes in geoducks of Vibrio pectinacida and a few other incidental cases. So could have easily been missed because it's not one of the ones that we monitor for that carefully. So it's a really exciting, interesting question to see where and when it is prevalent and how the stars are getting it. And also the origins of the Vibrio pectinacida and this particular epidemic are still not known, but I think now that we know the causative agent, we should be able to find out if it was some kind of a new introduction to our waters in 2011 or 2012 or 2013, or if it had been present in our waters and changed. And so both of those are sort of known roots of sort of a new pathogen showing up. And we don't have the answer to that, but I'm really confident that now we will be able to get an answer.
Your hand is up next.
You talked earlier about the pathogen for eelgrass. Does that pathogen attack all grasses? In other words, both the phylospadics and the outer rocky areas and the zostra in the sandier or shallow areas?
Yeah, that hasn't been, I think I heard your question. Sometimes it's a little hard for me to hear through the microphone, but I think you asked whether the Labyrinthula zosteri is present in the other seagrasses in our waters. It's sort of most prevalent in our Zostra marina, but also Japonica, which is an introduced zostra. And this is work that Morgan Eisenlord, was a former PhD student in my lab, did, and also detected in the phylospadics. We haven't really studied it in the phylospadics, and there hasn't been an indication that it's caused anywhere near the level of damage in the phylospadics that it's caused in Zostra marina. It's been most prevalent in the eelgrass Zostra marina. Yeah. Interesting. One of the reasons I'm here in Monterey is working at Hopkins Marine Lab with a couple of grad students who are working, one in Italy and one in Palau. And for two completely different species of eelgrass, they've been able to pick it up, causing lesions in those plants. So, and it is prevalent around the world in temperate Zostra marina, as well as now it's showing up in some of these other species as well.
Say in a vacuum, you were able to mitigate the disease, the pathogen levels in the eelgrass. How quickly do you think the like intertidal eelgrass meadow biomes would recover, if at all?
Wow, that's a great question. Boy, we sure wish we could do that. I mean, we've thought about various schemes, like, for example, in our lab experiments, we run our eelgrass plants through low salinity water, which tends to knock back the labyrinthial ostory because it doesn't do well in quite fresh water, whereas the plants will tolerate it. So we have thought about, just going out to some test sections and just going every low tide and spraying them with fresh water and just seeing. Uh, you know, uh, I can't claim that all the mortality that we're seeing, at least in the San Juans, is due to the labyrinthial ostory. There's also been a lot of just plain heat stress. We've had in the 2021 June heat wave, we had 100-degree temperatures in our intertidal and actually on my deck in False Bay. I mean, we'd never seen anything like that before. And so that kind of extreme temperature stress is just really doing in our intertidal plants. And so even if we were to take away the disease, I think we'd still have some problems, but the disease is making things a lot worse. So I hope that's a fair answer to that question.
Thank you.
Hi, Drew. Thanks so much for this. Do we know what it is about the rising temperatures that's making the Vibrio so effective at C-star wasting disease?
We actually, so Alyssa and Melanie have been running those experiments in BC literally this fall. So they've been exposing stars over a range of temperatures and trying to figure out, partly it's that the Vibrio grows faster at warmer temperatures, so it has an advantage under warmer conditions. And then there's a component of the stars being stressed under those conditions. And so we don't have the full answer to that yet. The other thing that's interesting about the Vibrio pectinacida is it makes a really nasty toxin. And so part of the damage that it's doing is the toxin that it's releasing. At least that's been reported from the scallop larvae. Yeah, and it's a sneaky one. One of the reasons why we had so much trouble identifying it. Normally, and you saw the pictures I showed of the eelgrass histology, the pictures looking into the cells of the eelgrass. I mean, we did all of that early on in the sea star wasting epidemic, and nothing showed up in the histology, which is why initially we thought it was a virus. But it turns out that for some reason this bacterium just does not show up in histology, and that was something that the scallop larvae people also reported. So it's got some new tricks.
Hi, Drew. I'm wondering about the selective breeding part of the Sea Star project that you mentioned, and I'm wondering if you've seen that individual basis resistance and how that might work in a lab setting on you actually making that long term.
Yeah, well, please understand that this is all kind of new because the pathogen has just been reported. It's just being, continuing to have a lot of confirmatory studies and all of that. And so there were over 250 stars in the experiments that our team worked on, control stars, and every one of those, sorry, 251 experimental stars, every one of those died. And so that's a little worrisome from the perspective of wanting to do selective breeding for resistance. Our thought is that, though, that those were pretty high dosages they were exposed to. So one of the projects that Alyssa is now carrying on with Melanie Prentice is using lower dosages to see whether some stars will pull through and there will be more evidence for resistance. But at this point, there's not been a lot of resistance seen in the sunflower star. And so, it's still unclear how well that's going to work. We can't believe that there's not some resistance, but uh, we don't have the answer to that yet. That's one of the reasons we're kind of interested in figuring out more about the leather star and what kinds of resistance genes it has, because it's not beyond the realm of, you know, conceivable to think about some bioengineering approaches as well, but hopefully that's not what's going to be needed. Yeah. The other thing that's underway, there's a lot of really exciting work, is phage therapy, is examining whether there are phages, which are viruses that eat bacteria. There definitely are phages in nature that will eat Vibrio pectinacida, and so is there some kind of phage therapy that can be implemented? A little hard to think about how that could be successful on a continental scale, but I have to say, if anybody's read my earlier book, Ocean Outbreak, I talk about the black abalone that were taken down by a Rickettsiobacterium, and a natural phage did come in and has conferred a lot of resistance under natural conditions. So it's not beyond the pale to imagine that happening.
Hi. So you talked about the success with the selective breeding and success with cryopreserved larvae and success in isolating the cryopreserved larvae in other species. Do you think that that's going to become standard practice in species conservation as we're going to start banking larvae, kind of like seeds in the ground in Siberia?
Oh, I think so. I mean, the first meeting that we had, we literally, Nature Conservancy brought in the white rhino people and the oyster breeding people and the salmon selective breeding people to actually say, do you think we can be successful in conservation breeding for the sunflower star? And once they heard that the larvae could be cryopreserved, they're like, yeah, we do think this can be done, because you do have a way to be able to biobank, in the worst case, which we hope is not going to happen, where there's a new strain of the Vibrio that comes and decimates them all the way up to Alaska. We don't think that's going to happen, but if that did happen, we would at least be ready for that, in that animal would be cryopreserved and biobanked. And so there now are several other species of our stars, including Pysester brevispinus the larvae of that one have been cryopreserved as well. So yeah, I think it's going to be an important tool in species recovery.
Follow-up question. In that case, are there being efforts made then, if it's been so successful across multiple species like that, I'm sure you guys are doing research, but are you guys maybe doing research on that to put together like an SOP so that can become standard practice across species conservations? Like, say some people aren't as privileged as to have everybody show up to tell them about it in person. Like, what efforts are being taken to like pass on this knowledge to other people?
Yeah, it has actually been, for me, because I've worked in marine ecology for my entire career, and I've watched a lot of diminishment of species, like, for example, with corals, and some other taxa. This has actually been really hopeful to see the way such a large team has come together, the way the aquaria, all the major aquariums have really pitched in and helped and developed methods and information from other species recovery efforts. For example, the pinto abalone, that's a big one in Washington waters, and the development of standard operating protocols in those cases were basically used to develop some of the early approaches in the sunflower star recovery program. And so, yeah, those methods definitely will be reproduced and shared widely. Yeah. Not by me, because I didn't do any of the cryopreserving of the larvae, and I don't actually know how to do that, but yeah.
That's really interesting. I read a couple of the papers from the pinto abalone studies that you're talking about. That's really interesting. I'm quinol, so like that, the ocean acidification and the weakening of the hinges really affects like our entire food pathways and everything. So that's something I've been following really closely. So that's really
Yeah. Yeah. Thanks for that.
Thank you.
Any last burning questions? Keep loud.
This question is mainly about like starfish physiology. When a starfish loses a limb, and say the starfish is at the end of its life, and I did hear that at times they can asexually reproduce through that limb and create another starfish. At what point, like what age will that clone of that starfish be?
I had a little trouble hearing your question. I'm not sure. It had to do with arm loss, but I didn't quite get the last part of your question. Maybe Clarissa can help.
It was the age of the sea star that would be formed from that clone.
She said that when sea stars will drop limbs towards the end of their lives and are like getting close, some of them can asexually reproduce. She was wondering when that would happen for the clone to exist. Yes, to come into. Yes. Yes.
And, like,
Okay.
What like cells will be carried over? Like will the age of the main original starfish be affecting the clone of that starfish?
Oh, would the age of the original starfish that asexually reproducing created the clone, would that affect the life of the clone and like how it exists? Or, yes.
I don't actually know the answer to that. So, because not all stars autotomize or drop their arms. So there are some species that is a normal part of their life cycle. They drop their arms and can completely regrow another star from them. Sunflower stars are not one of those. When they drop their arms, usually the arms do not regenerate a new star. They can regenerate new arms as long as the central disk is still there. And so that's why sometimes you'll find a star that's got like an arm that's just starting to grow back. So I've never worked with the species of stars that routinely drop arms in asexual reproduction as part of their reproductive mode. I'm not sure that really any of ours do that regularly. Yeah, so I don't know the answer to that.
Well, thank you again, Drew, for your time tonight to share.